Today let us discuss about primary standards and secondary standards. So what do you mean by a primary standard? A primary standard is a highly pure, stable, non-hygroscopic substance with a known molecular weight. Whereas a secondary standard is a substance whose purity is not high enough to be used directly. Hence, its concentration must be determined and standardized against a primary standard solution. Here, a primary standard is a highly pure, stable and a well-characterized substance used to calibrate other standards. While a secondary standard is a substance or a solution whose concentration is determined by titration against a primary standard and it is used for routine analytical work. Primary standards must meet strictly the purity, stability and reactivity criteria, whereas secondary standards are less pure and may change over time, which requires a periodic recalibration. So let us see about the definition of a primary standard. A primary standard is defined as a substance of high purity and stability with a known exact composition used to standardize other solutions or instruments. The key characteristics of a primary standard are as follows. So the primary characteristics are high purity, stability under various conditions including temperature and moisture, non-hygroscopic, high molecular weight and easily obtainable and purifiable. So what do we mean by high purity? The primary standards should have an extremely high purity, typically 99.98% or higher, meaning the total amount of impurities should not exceed a very small percentage like 0.01 to 0.02% to ensure accurate and reliable analytical results. In simple words, if any impurities are present in the primary standard, the total amount of impurities should not exceed 0.01 to 0.02%. This exceptional purity is essential because impurities can significantly alter the measured concentration of a solution, leading to incorrect calculations in titrations and other chemical analysis. Also, high degree of purity is crucial because a primary standard is a pure stable substance of known composition which is used to accurately determine the concentration of other less pure substances through the titration. So let us see why purity is critical. Purity is critical to determine the accurate mass to volume ratios. A high purity primary standard allows for precise weighing of the substance, which is then dissolved to create a solution with a known accurate concentration. Also, to provide a reliable standardization. The accuracy of primary standards is crucial for calibrating secondary standards and analytical instruments, since any error in the primary standard will be propagated throughout the subsequent analysis. Prevention of interference. Impurities present in the primary standard may interfere with chemical reactions being studied, which potentially leads to incorrect endpoints or misleading results. Primary standard should be stable under various conditions including temperature and moisture. Primary standards must be highly pure, exceptionally stable under both storage and usage conditions. It should possess a low tendency to absorb moisture, that is hygroscopicity, and have a high equivalent or molecular weight to minimize weighing errors. They also need to be readily available and non-toxic and soluble. Stability in this context means The substance's chemical composition and physical properties like mass should remain constant over time and storage conditions as well as usage conditions. This prevents change in concentration or mass. So why stability is crucial? The primary purpose of a primary standard is to act as a reference material to accurately determine the concentration of other solutions or to calibrate an analytical equipment. If the primary standard itself is unstable, its mass or concentration will change over time, making it unreliable for critical functions and leading to errors in subsequent analysis. Non-hygroscopic. A non-hygroscopic substance is the one that does not readily absorb moisture or retain moisture from the atmosphere. So here the primary standard should have a very low tendency, that is low moisture affinity, to attract and hold water from the surrounding air. Stability. Their inherent resistance to moisture keeps their composition and behavior stable, preventing changes that could occur if they absorb water. So why does this matters? Product stability. For products like pharmaceuticals or certain materials, being non-hygroscopic ensures that these products do not become degraded or unstable due to humidity. And consistent performance. In manufacturing materials, they should maintain a defined property even when exposed to different environmental conditions. And a high molecular weight. Primary standards should have a high molecular weight to minimize weighing errors, making it easier to prepare solutions of accurate concentrations for analytical chemistry. A higher molecular weight means that any small unavoidable error in the weighing process becomes a smaller percentage of the total mass, leading to a more precise and reliable measurement of the substance. And also, the primary standard should be easily obtainable and purifiable. So, easily obtainable and purifiable means the substance is readily available and can be purified easily to obtain a highly pure material for accurate analysis. So, what are the steps to obtain a or to prepare a primary standard solution? So step 1 will be to purchase a highly pure standard. That is, purchase a commercially available chemical that is known to be a suitable primary standard. Step 2 will be to dry the substance. Dry the primary standard in a drying oven at a specific temperature until it reaches a constant weight. Step 3 is store in a desiccator. Store the dried primary standard in a desiccator to prevent it from absorbing moisture from the air. Step 4. Accurately weigh the material. So using an analytical balance, weigh the exact amount of the primary standard needed to prepare the desired solution. And step 5 is dissolve the substance. Transfer the weighed material into a clean volumetric flask and add a small amount of the solvent, which is usually distilled water or deionized water, to dissolve it. Step 6 is dilute to the mark. Add more solvent to the volumetric flask carefully, bringing the meniscus to the calibration mark to achieve the precise and final volume. Step 7. Mix thoroughly. Cap the flask and invert it several times to ensure that the solution is completely mixed and uniform. The resulting solution has a precisely known concentration derived directly from the accurately weighed primary standard and then can be used for titrations or other quantitative analysis. So what are the uses of a primary standard? The primary standard is used to prepare standard solutions of known concentration. It is used to standardize or calibrate secondary standard solutions, and the examples include potassium hydrogen thalate, sodium carbonate, or pure metals like copper or magnesium. Moving on to a secondary standard. So what is a secondary standard? A secondary standard is defined as a substance that is not stable or pure enough to be a primary standard, but it is used for routine analysis. Its exact concentration must be determined through titration against a primary standard solution. So what are the key properties of a secondary standard? So lower purity. It has a lower degree of purity and may contain impurities or water. Less stable and more reactive. It may react with atmospheric components affecting its concentration over time. Standardized by titration. Its exact concentration is determined by reacting it with the primary standard. And also it should be more convenient and cost effective. Since these secondary standards are used for routine procedures, they are less expensive and should be readily available than a primary standard. So examples of secondary standards include in acid-base titrations, compounds like sodium hydroxide and hydrochloric acids are secondary standards because they are hygroscopic and absorb moisture or CO2 from air. Hence, these cannot be used as a primary standard. And in redox titration, potassium permanganate is used as a secondary standard. Thank you.